All right, here we start section 7.2. But before we go into the second part of microbial metabolism, I'd like to take just a minute and review 7.1. So, which of the following mechanisms of enzyme control blocks a reaction catalyzed by an enzyme by the binding of a product to a regulatory site of the enzyme? All right, so your options are enzyme repression, competitive inhibition, enzyme induction, non-competitive inhibition, or none of those are correct options. All right, hopefully you will recall that we actually didn't talk about enzyme repression, nor did we talk about enzyme induction. So you are left with B, D, or E as options. And if we look at the figure 7.6, where it looks at the two forms of control mechanisms, you have competitive inhibition and non-competitive inhibition. The question read, which one acts through the regulatory molecule, or a molecule binding to the regulatory site, and this is that regulatory molecule, and so therefore the answer is going to be non-competitive inhibition. All right, then moving on to section 2. This section really talks about energy, and not necessarily the production of energy, but just the concept of cellular energy. And so there are just two objectives for this section. It's quite short. What are redox reactions? So understanding the idea of reduction and oxidation of different chemicals or molecules. And then, what are the three components of ATP? And this will be seen in figure 7.10. All right, so energy, you hopefully know, is or can come directly from light. All right, so if an organism is photosynthetic, they utilize the energy from light. But a lot of times we have cells that are not photosynthetic, like animals. And so we're going to have to utilize energy through the means of pulling in chemicals and utilizing chemicals. And so energy is going to actually be stored physically in the chemical bonds. And the chemical that we're talking about primarily for energy production or storage in cells is going to be through ATP. All right, so energy is stored in ATP through those bonds of the adenosine triphosphate. And normally, energy is going to drive cell transactions, the reactions, the chemical activities within a cell. All right, so one of the two major concepts in this section is the oxidation and reduction reactions, or sometimes just referred to as redox reactions. All right, and so this chapter or section starts off by saying some atoms and compounds readily get or receive electrons and participate in the oxidation, the loss of electrons, or reduction, the gain of electrons. All right, so oxidation is the loss of electrons, and so when a compound loses electrons, it is going to be oxidized. And the reduction process is a gain of electrons, and so when a compound gains electrons, it is reduced. And it's kind of backwards, all right? So you gain electrons, but it's reduced, all right? Gaining and reduction are opposite of each other. But if you recall that electrons have a negative charge, and so you're actually reducing the overall charge by adding an extra negative to it, all right? And that's the electron. So if a compound gains an electron, it's reduced. And you have this reaction of oxidation and reduction, and they're usually just called redox because in almost every case, these reactions occur simultaneously or within each other, because you don't want free electrons just running around all by themselves in the cells, right? That's not safe for the components. So redox reactions are paired up because they occur together. All right, so concept check. In a reduction or a redox reaction, loss of electrons is phosphorylation, oxidation, fermentation, reduction, or none of these? All right, the correct answer is oxidation. So losing electrons, it's oxidized. Gaining electrons, reduced, or reduction, because the overall charge is reduced. All right, then ATP. All right, ATP, again, is where cells are going to store its energy. And it's called adenosine triphosphate. And so what you have in this figure is the nitrogenous base, the adenine, and then you have the ribose sugar. So this actually is very similar to a nucleotide you might find in RNA, right, for the ribose. And then you have this one phosphate group, and you can have another phosphate group that locks onto the first one. And if that's the case, you have adenosine diphosphate. Add a third phosphate, then you have adenosine triphosphate. Right, so it's a chain of these phosphates. And the removal of the outermost or terminal phosphate releases energy, and that's what you have here with these little symbols here. All right, so when that phosphate jumps off, the energy that is stored between the bonds that are connecting these molecules is released. And you can then, or the cell can utilize that energy. All right, and that's actually the end of section 7.2. So it's relatively short, just those two major ideas or topics of redox reactions and ATP. So it's very important that you remember the three components of ATP. That's one of your chapter objectives, right? And then in that concept of remembering the three components, where's the energy stored? The energy is stored between or within those bonds that are between the adenosine or the phosphate groups. And as you move out, just another side piece of information, as you move out, you create more energy. So adenosine triphosphate has more energy than adenosine diphosphate simply because the connection between the third and the second phosphate group stores more energy than energy that is stored between the second and the first phosphate group. So adenosine diphosphate is also a form of energy, but it's just not as powerful as adenosine triphosphate.